Hi everybody. Let me discuss about major histocompatibility complex. Major histocompatibility complex was first detected as the genetic locus encoding the glycoprotein molecules. So glycoprotein molecules also act as a transplantation antigen. So they are responsible for the rapid rejection of uh, grafted tissue. So the grafting between genetically non-identical twins that may be rejected occurred in 1930 was the first to identify the antigen responsible for allograft rejection. He demonstrated two blood group antigen 1 and antigen 2 in mice. Antigen 1 was found in all type of mice, while antigen 2 was found to be a certain species only. Transplantation antigen coded by a closely linked multi-allelic cluster of genes like major histocompatibility complex. So this was named as H2 complex. Histocompatible antigens are new surface antigens that induce immune response to an incompatible host that results also known as allograft rejection. The MHC in a human is known as human leukocyte antigen. In a human, these alloantigens are present on the surface of leukocytes and are called HLA, and the set of genes encoding for them is named as HLA complex. Uh, Carbohydrate antigen of erythrocytes and glycoprotein antigens of cell membrane are the two types of major transplantation antigens of humans. Snell, Dataset, and Penserev 1980 were awarded the Nobel Prize for their work on major histocompatibility complex and genetic control of immune responses. MHC antigen that include induce immune response to an incompatible host, resulting in allograft rejection. So the image also depicts uh, stratum of chromosome 6. In a human, the HLA complex of genes is located on stratum of chromosome 6 containing several genes so that are critical to immune functions. The HLA complex of genes is classified into three types, like the stratum of chromosomes 6 also contain these three types of MHC molecule, class 1, class 2, class 2, class 3 molecules. So class 1 molecules also contain the HLA region, HLA-B region, and HLA-C. Okay? So HLA-A, HLA-B, HLA-C. Then class 2 also situated in the D region. So this class 2 MHC also known as DPTQDER. These are three components situated in the D locus. Third one, class 3 MHC molecule. So class 3 MHC molecule also complement of there is called complement loci. So that locus also include complement proteins. So that complement proteins also involved in alternative pathway and classical pathway. So these are it for C4A, C4B, C2A, C2B, then factor B, then tumor necrosis factor alpha and beta, etc. So this is class 1, class 2, class 3 MHC, class 1 HLA-A, HLA-B, HLA-C, class 2 HLA-D or DQ. Q and DP. All of these are present within HLA-D region of HLA complex. Class 3 complement losing that encodes C2, C4, factor B of complement system and tumor necrosis factor of alpha and beta respectively. A locus mean on the position where a particular gene is located. HLA loci are usually multi-allelic. That means the genes present on the locus can be any one of several alternative forms. There are 24 alleles at HLA-A locus, and HLA-B locus also contain nearly 50 alleles. Each allele express for a distinct antigen. HLA system is phylomorphic, and every person inherits one set of HLA gene from each parent. HLA genes encode a variety of enzymes and structural molecules essential for the activation and functions of T and B cells. The genes encode MHC proteins that are classified into three groups or classes known as class 1, class 2, class 3 molecule. The first one, class 1 molecule. These are glycoproteins found on the surface of virtually all liquid cells in the body. Class 1 proteins were encoded by HLA-A, HLA-B, HLA-C loci. Approximately 20 proteins, approximately 20 proteins allelic, 20 proteins of encode the allelic genes at the A locus and 40 H B locus and H B C locus. The complete class 1 is composed of a transmembrane glycoprotein of 45,000 Tollin, which is non-covalently associated with beta-2 microglobulin. The heavy chains, like that of an immunoglobulin molecule, has a variable and constant region. The variable region is highly polymorphic. The polymorphism of this molecule is important in the recognition of self and non-self. The polymorphism is used to recognize whether the molecule is self or non-self. If the molecule is non-self, then the MHC system also react against that non-self molecule. So this image also depicts class 1, class 2 MHC molecules. The class 1 molecule, then alpha 1, alpha 2, alpha 3, and beta microglobulins. So the beta microglobulins are non-covalently associated with cell membrane. Then class 2 molecules, then alpha 1, alpha 2, and beta 1, beta 2. So then both associated with a cell membrane. Then class 2 molecules, glycoproteins, unlike class 1 proteins, they have restricted tissue distribution. It chiefly found on macrophage, B cells, and other antigen processing cells such as dendritic cells of spleen and Langerhans cells of the skin. Their expression on their cells, endothelial cells, can be induced by a gamma interferon. They are highly polymorphic glycoproteins composed of two non-covalently associated transmembrane glycoproteins, molecular weight of about 33,000 Tollin and 29,000 Talton respectively. Class 2 proteins are encoded by the genes of HLA-D locus. These locus retains the control of immune responsiveness, and different allelic forms of these genes confer striking differences in the ability to mount an immune response against a given antigen. The class 2 MHC locus also includes genes encoding proteins involved in antigen processing. Class 2 proteins are primarily responsible for the graft versus host response and the mixed leukocyte response. Class 3 MHC locus encode complement protein C2, C4 of the classical pathway, a preferring factor B of alternative pathway, and several cytokines. So the class 3 MHC, also known as complement loci. So the complement loci encode the complement proteins C2, C3, C4, C5, 6, 7, 8, 9 respectively. So these complement proteins also, which was mainly involved in the uh, rejection process. So mainly involved in the rejection process. So that also act as a perforin proteins. So that have a great immunological importance. Then the next biological importance of MHC. Transplantation antigens are responsible for antigen recognition by T cell receptor. T cell receptor only recognize the antigen presented by MHC molecule on another cell, the antigen processes. The TCR is specific for antigen, but the antigen must be presented on a self-MHC molecule. Peptide antigen associated with class 1 MHC molecule are recognized by CD8 cytotoxic T lymphocytes, whereas class 2 associated with peptide antigens are recognized by CD4 helper T cells. Thank you. Thank you for watching this video.